Hey, Hammy here. Here with the first video of Chapter 7, where we begin to dive into what exactly is the genetic material and what is its structure, how does it copy itself, and how does it work. And so the first part of this video series here, we're going to begin to look at the central dogma of biology and also one of the experiments that first started us down that road in the 20th century to discovering that the genetic material was DNA. Okay, one of the first things we need to mention is it's very important in molecular biology, we talk about the central dogma. Okay, so that's like the key tenet, the main thing. Like everything else hinges on this, and that's this idea of deoxyribonucleic acid, okay, and how that is the genetic material of the cell. It stores information and is passed on from generation to generation and can be copied very accurately over and over and over again. One of the main questions we'll look at this chapter is how do instructions in the DNA, and remember, where do we find DNA in eukaryotes? Okay, if you look down here, right, they're in the nucleus. Here's the little chromosomes, okay, in the nucleus of the cell. Well, they have the instructions or the code for making the proteins, because remember, proteins are used as enzymes, so all those chemical reactions that are taking place, they're used for building materials. But how do we get from the blueprints in the nucleus out to the ribosomes, where the proteins are actually built, okay, out here in the ribosomes? Okay, so how that's done is through another nucleic acid called ribonucleic acid, hence the name RNA. And in a later video, we'll look at kind of the difference between RNA and DNA and why their names are kind of different. We're going to look at this chapter. First, we're going to start with DNA. How was it discovered? And some of the famous scientists and names that go with that. And then the second part of the chapter, we'll look at how DNA is copied, and also how DNA is taken from the nucleus in the form of RNA out to the ribosome to make a protein. Okay, so this idea right here is the central dogma of molecular biology, that who we are starts as DNA, is coded into RNA, which is then made into a protein. And that's how it is expressed as our phenotype, we would say, from last chapter. Okay, you remember last chapter, we talked about Gregor Mendel and how he did a lot of work with inheritance, okay, but he had no idea about DNA or what exactly carried those genetic factors in the cells. Okay, in the early 1900s, in the early 20th century, the search kind of began for what is this genetic material. And part of that started with a guy by the name of Frederick Griffith, who was a British scientist who did a lot of work studying pneumonia. Okay, pneumonia is caused by a bacteria, and he found that there were two strains of pneumonia. One that was called a smooth strain, which we'll often abbreviate with a capital S, and a rough strain, which we'll often abbreviate with a capital R. Okay, and the smooth strain, when he grew them on Petri plates, you can see down here, they had kind of this kind of capsule, or they look kind of like bigger, smoother, I guess. Whereas the rough strain were just individual cells, and they didn't have that like clear, smooth capsule part around the cells. And when he injected these into lab rats, okay, he noticed several things. First off, the living strain is what we call virulent. In other words, when he put the living S strain into the mouse, what happened to the poor mouse? Okay, the mouse got pneumonia, and after a couple of days, the mouse died. Okay, when he did a culture, okay, in the blood, in the heart, in the lungs, okay, he could find living S strain cultures in the tissues. Okay, made sense. The bacteria got into the mouse, and these little capsules or slime coats prevented the immune system from being able to destroy them. And so they caused pneumonia, caused death. The R strain was harmless. Okay, why? It didn't have that coat. So when he injected the R strain into the mouse, okay, the mouse is happy, because the immune system can fight it off. After several days, he took a blood sample, and there was no bacteria. The immune system had fought him off. So S strain caused death, R strain harmless. The mouse lived. Then he started kind of doing some experiments with that, kind of extending that. The first one, he took the S strain, and here's a little flame here. Here, he added heat. Now, what would heat do to the bacteria? It would kill the bacteria, right? So when he put heat-killed S strain pneumonia bacteria into the mouse, what happened? The mouse lived and was very, very happy. Okay, and he didn't find any bacteria in the tissue because the S strain could cause disease, but if the bacteria was dead, then they did not cause disease, which kind of makes sense, right? But the fourth thing he did kind of puzzled him a little bit, because when he mixed the heat-killed S strain and mixed them with living R strain, he thought, well, R strain, mouse lives. Heat-killed S strain, mouse lives. So the mouse should live, right? Wrong. The mouse died, and an interesting thing happened. When he cultured the blood and the heart samples, he found living S strain. So on your notes, heat-killed S plus live strain. Okay, the mouse died. And you can write in there that he found living S strain inside the mouse tissue. Now, what in the world happened here? Okay, Frederick Griffith's hypothesis, or the word he used, was transformation. In other words, he said there was something in that heat-killed S strain that changed, that permanently changed the harmless bacteria, the R strain, into the disease-causing strain, or the S strain. Okay, he said there was a transforming factor, okay, that caused, okay, this change to happen. Okay, and whatever that change would be is the genetic material. Okay, so he was looking for what was in that transformation juice, he called it, that the R strain, the living R strain, what did they pick up? What cellular components did they pick up from the S strain to transform them into the disease-causing bacteria? Okay, then the race was on. When people realized if we could just figure out what was in this transforming juice, then we would know what the genetic material was. And a lot of people knew there'd be a lot of fame and fortune if you could figure out what all these genetic material that Mendel was talking about, how exactly are genes passed on from generation. Okay, so in the early 20th century, 1928, when this was published, this really got people, and people started repeating this experiment over here over and over and over again, and they took that, tried to find that transformation juice and tried to figure out what is that genetic material, and the race was on.